225 General Microbiology with Lab by Denise Hernandez, due July 21st, 2025. And here it is. Hello, my name is Denise Hernandez, and I am conducting a microbiology with lab exercise on aseptic technique and culturing bacteria in broth. The purpose of this experiment is to reactivate the lyophilized bacterial culture and subculture in it, nutrient broth, maintaining aseptic conditions to avoid contamination. Here we have all my tools that I've used for this experiment. And what I have done for this experiment, before starting, I have washed my hands thoroughly with soap and water, and I have put on my personal protective equipment, which is including the gloves, the safety goggles, and my lab apron. Next, I have disinfected my work surface and the culture to lab using a 10% bleach solution. It's important to allow the disinfectant surfaces for about 2 minutes. I have labeled here the sterile broth tube with unknown organisms, and here it is. And today's date, which is July 21st, 2025. Using a greased pencil, I have reactivated the lyophilus and bacterial culture. First, I have removed the metal band, which is this right here, from the culture vial, and I have discarded it to the side. Next, I have took a pigment from its wrapping with my dominant hand. Using the pinky finger, I have removed the cap from the broth tube without sealing it down. I have flamed the mouth of the broth tube to sterilize it. And after each step, I've been flaming the mouth of the tube just to avoid any contamination of any sort. I have used a pipette to draw up 1 milliliter of broth. I have flamed the mouth of the broth tube again. Then I have been replacing the cap back on it and setting this into the side. Now I have opened the lyophilized cultural vial and dispensed the broth. I have mixed the culture gently by squeezing and the pipette water to avoid introducing any air bubbles. I have mixed the culture gently. Finally, I have flamed the mouth of the broth tube once more and replacing the cap, and will incubate for about 24 to 72 hours. I will then disinfect my area with alcohol. With this particular assignment, this concludes my lab exercise for this step of this experiment that I have processed for this assignment, for this technique and exercise for the culturing bacterial and broth. Thank you everyone for watching.